Three, two, one.
Cell cycle!
Alright, so a general overview of the first phase or inner phase of our cell cycle. It starts off with G1, which is growth. So that's where a cell is going to spend some time, it's going to be growing, it's just going to be doing what a cell does. And then right before it goes into its S phase, we have the G1 checkpoint, which makes sure that the DNA isn't damaged and that the cell is ready to commit to cell division. And then we hit S phase, which is the growth and DNA replication of that cell. And then we move on to G2 phase, which is the growth and final preparation for the cell division. So that's where we've replicated it, it's getting ready, it's copying the organelles, and it's about to divide and go into mitosis. And it's going to also have the G2 checkpoint, which is where if the DNA is, it's making sure the DNA is replicated properly right before it's about to divide. And the cell spends a majority of its time in interphase before it divides.
Okay, after it gets past the checkpoint and moves into cell division, the first phase of mitosis is prophase. So in this phase, the DNA will condense and form chromosomes. It does so by wrapping itself around histones and then forms rosettes of chromatin loops in order to condense, form a chromatin loop that twists and forms the chromosome. The chromosomes have kinetochore microtubules on the kinetochore that is in the center. And then the spindle fibers will attach there and move them towards the center of the cell in the metaphase plate, transitioning it into metaphase.
The third phase is anaphase. So anaphase begins when the centromeres split, and then the cohesion proteins are removed from all the chromosomes, and then the two sister chromatids are pulled apart to opposite poles. And the two forms of movement that make this happen is anaphase A, which is when kinetochores are pulled towards the poles as microtubules shorten. Then anaphase B, when the poles move apart as spindle fibers slide past each other. And then we have telophase. So telophase is when the spindle apparatus disassembles. The nuclear envelope forms around each set of sister chromatids, and then the chromosomes begin to uncoil, and then the nucleus reappears. The nucleolus reappears in each new nucleus.
After that comes cytokinesis, the last part of the cell cycle, and that is when the cell splits via the actin contracting, and then you have two daughter cells. But also, we took all this information, learned more about it, and then we applied it in the lab, primarily by looking at onion root tip cells. And at the beginning, we hypothesized about the time it takes in each part of the cell cycle. So for interphase and then the four steps of mitosis, we predicted the time for interphase. We knew it took the longest, so we predicted about 23 out of 24 hours. And then we split up the increments of time remaining equally for the four steps of mitosis. And then in the lab, we counted onion root cells. And then we identified what stage of the cell cycle they were in, counted them all up, tied them up, and then put them in percentages, and then we multiplied the percentage by 24 hours in order to get a more accurate representation of how long each part of the cell cycle takes. And we concluded that we overestimated the time it's in interphase, where it's more about 900 minutes rather than 1300. And then the parts of mitosis take about 150 minutes each, give or take. But yeah, this is what we learned, and we really had fun. Thank you.